Hi students, so today we are moving on to a new chapter. The name of the chapter is Biotechnology, Principle and Processes. It's a very interesting chapter. So biotechnology as a branch of biology. So what is biotechnology? Technology in biology, which means that something we are going to make for our benefit, for the benefit of the human beings using living organism. That is what we call it as what? Biotechnology. As I am telling you the definition, you can make out. Ancient biotechnology. Biotechnology, it's not a new branch. It's already present from years, decades ago it was present. Like fermentation, when it was discovered, when we are finding out that process, the fermentation process is also a biotechnological process, because in fermentation what is used? Yeast or the bacteria is used for breaking down the carbohydrate stuffs, the sugar stuffs, and they are releasing what? They are producing something like alcohol, carbon dioxide. So anaerobic respiration is taking place with the help of this microorganism is also what? A part of biotechnology. So ancient days itself we can find this field is there, biotechnology is there. Nowadays advanced one, modern biotechnology, wherein what we are going to do is that we are going to cut, paste, with DNA we are going to play and make new things which is useful for mankind. That is what this chapter is going to deal with. In that, the main portion what we are going to discuss is rDNA technology. What we are going to discuss? rDNA. So DNA, you know what is rDNA? DNA is deoxyribonucleic acid. So what is R here? R is written with small letter. R is what? Recombinant. It's not the natural DNA which is present in an organism. I am going to make a DNA with pasting something into the natural one. Okay, that's why it's not the original one, it's a recombinant one, it's a different one, because I am going to make it. Okay, so that one, that branch we call it as what? rDNA technology. So that is the one which we are going to mainly focus on this chapter. So rDNA means recombinant DNA. So naturally, imagine this is a cell, this is a nucleus, inside there is a DNA. That DNA is normal. In case if I am making some changes in that DNA by cutting some sequences and introducing some sequences into it, it becomes what? A new one, modified one, right? So that one we call it as what? We call them as rDNA, recombinant DNA. Okay. So this is what we are going to discuss. This rDNA technology can also be called as cut and paste. I am going to cut some parts of the DNA from the original one, and then I am going to paste something new into it. So cut and paste technology is also rDNA technology, another name. Other name for the same is your genetic engineering, genes. So DNA is what it is made up of? It is coding for different genes, isn't it? So genetic engineering. Engineering means I am going to engineer, means make new DNA by cutting and pasting. So that genetic engineering is another name for what? This rDNA technology. So rDNA technology are having different names. So what are the different names? I told you as such. One is rDNA technology, same only. Cut and paste technology. I am going to cut and paste. Okay. When I am going to cut and paste these things, which means I am going to cut the gene sequences and paste something else into it. So it can also be called as what? Genetic engineering. So these are the different names of the same rDNA technology, recombinant DNA technology. That is what we are going to mainly focus on. Before moving into this rDNA technology, we need to know some facts regarding biotechnology. The first one is your definition. What does biotechnology? Biotechnology deals with techniques, whatever techniques it is, whatever processes, in which what we are using, we are going to use living organisms. So with the help of living organisms or enzymes, I am going to produce what? Different products. It can be an antibiotic, it can be a vaccine, it can be some pest resistant or disease resistant variety. All these are what? Some things which we are going to produce. Okay, some products we are going to produce with what? With either the living organism or the product which we are getting from the living organism. It can be an enzyme, it can be a hormone, a juice secreted by some microorganism or any other living organism is called as what? Biotechnology. So it's all involving techniques which are using living organisms to make something, to make some products which is useful for mankind, okay, or humans it is useful for. So that one is biotechnology dealing with. Now The term biotechnology was given by Carl Ericke. So, this information you need to know, one word. And European Federation of Biotechnology, EFB, full form is what? European Federation of Biotechnology, has given a different definition for the same. So, what is the definition? Biotechnology is integration of natural science, basic science, okay, natural science and organisms, the different organisms or the cells or the parts of the cell, the cell organelles, protoplasm, whatever, all part of the cells, or something like living organism, some molecules we are making, like molecules of proteins or fats or carbohydrates, we are making molecules. Analogs means like living organism, I am going to create something, okay, it will be having molecular structures which is resembling some living organism. So, that is molecular analogs. For why we are doing all these things, we are using these natural science, living organisms or molecular analogs, why we are making? In order to provide us with some products or in order to serve us in fulfilling some requirements, okay. So, we are making all these things for what? For making some products or services, okay. So, this is what is dealing with, biotechnology is dealing with. Just a definition, you just read it all, right. Now, principles of biotechnology, two principles are there, okay. The first one we have already discussed, what is that? It is genetic engineering, okay. So, what is genetic engineering? Techniques to alter the chemistry of the genetic material. Genetic material can be what? It can be DNA or it can be RNA. DNA means what? Deoxyribonucleic acid. RNA means what? Ribonucleic acid. To introduce these into host organism and change the phenotype of an organism. Listen, so this is a cell, okay, an egg cell, imagine a fertilized egg cell. In that there is a nucleus. What is present? The nucleus is present. Inside there is a nuclear. Inside you can find the DNA, the genetic material, okay. Now, what we are doing is that I am taking out the DNA, okay. After taking out this DNA, I am cutting one segment. What I am going to do? I am going to cut one segment, and then what I am going to do is that then I am going to introduce a new segment into it, okay. So, I am, after cutting this one, this is gone. Next, what I am doing? I am introducing a new segment. Here I have introduced another segment which is having some property, like they are producing some antibiotic or they are producing some vaccine. That gene is coding for producing some vaccine, or the gene is coding for disease resistance, that is being introduced into this, right. Now, what we are going to do? I am going to take this DNA and then put it into here. Imagine that I am putting this DNA which I have modified and I am going to put it here into the seed. So, when this seed is growing up, if it is pest resistant, what the gene will be coding for? Pest resistant. So, that plant will be resistant to pest. So, I have created something new with the help of what? Cutting and pasting, with the help of genetic engineering. So, this one we call it as what? Genetic engineering. This seems very simple, but it is a very complicated task. We will be discussing in detail just for understanding the definition. Okay. So, genetic engineering, techniques to alter the chemistry of the genetic material. Previously, all read it was nothing new. Inside, what I have done? I have taken something and introduced what? Some other gene into it. Okay. Introduce these into host organism. So, now phenotype will change or not? Phenotype, the external character can change. Okay. Like if I want to produce a dwarf variety, the gene which is coding for dwarf is introduced over here. Okay. So, this is the DNA, right. Now, in this DNA, the gene which is coding for what is that? Dwarfness is introduced over here. Now, this gene, if I am introducing and making a plant, then that will be what? Dwarf in nature, which was not previously present. But since I have introduced that gene, that plant is turning dwarf. Dwarf means what? Short. Okay. So, dwarf varieties and all can be created with the help of what? Genetic engineering. So, genetic engineering, I am changing the DNA sequence. I am taking out some part and there I am introducing some part which is useful to me to make different varieties. So, that one we call it as what? Genetic engineering. That is the first point. Second one is that maintenance of sterile ambience. What do you mean by the term sterile ambience? Sterile means disease free, germ free environment. So, making all these processes possible, the biotechnology has to work out means you want what? You want a sterile environment. There should not be any other microorganism other than the microorganism what I am using. Then only the product will be fruitful. Otherwise, it will lead to some contamination and I will not get my result. So, in order to maintain sterile condition, germ-free condition, there also what is playing a major role? Biotechnology is playing a major role. So, microbial contamination free in chemical engineering processes to enable growth of only the desired microbe. I am allowing only the desired microbes to grow, so that any product what I am going to produce, I will be getting it. Imagine this one is a place where I am culturing penicillin. What I am considering? Penicillium notatum. Penicillium notatum. It is producing what? It is going to produce penicillin. You know what is penicillin? It is an antibiotic. Now I want to make many penicillin. Okay, so what I am going to culture there? Penicillium notatum. Okay. Now when I am growing this Penicillium notatum, if some other microorganism come and take up here, then I will not be getting the penicillin. Whether I will be getting it? No, isn't it? So in order to prevent that processes also, techniques are there. Okay, in order to resist the other microorganisms to grow. So that techniques are also dealt where? It is dealt in biotechnology. So maintenance of sterile ambience, that is in chemical engineering processes, which allow the growth of only desired. Desired is what? Here example I told you, Penicillium notatum in large quantity for the manufacture of biological products like antibiotics or vaccines or enzymes we are going to produce. I am allowing only my desired variety to grow and I am not allowing other microorganism to get contaminate there. Okay, so that is also dealt in what? Biotechnology. So the two principles of biotechnology, when it is asked, you have to explain what is genetic engineering. Second, what you have to say is that maintenance of what? Sterile ambience, sterile atmosphere, sterile environment. Okay. Now moving on to the next one is some facts. Traditional hybridization. What is traditional hybridization? Traditional hybridization, you know, one plant is there. Imagine this is plant A. Okay, so one is plant A and another one is plant B. In this plant A, what we are doing is that one of the flower is there. In that I am removing the anther. What is it removed? So this is plant A and this is plant B. We have discussed already hybridization. So plant A and plant B, one is high yielding, another one is disease resistant. Okay. Now what I am going to do? I am going to cross them. Okay. So pollen grains from one B is collected and dusted over the stigma of A. Now what will happen? The pollen grain will go and fuse, a new plant will be formed which will be having both what? High yielding as well as disease resistance. But sometimes what happens is that this B is having disease resistant, along with that there will be some other complaints like some other things which is not that, like maybe the color is not there or it is producing some other poison which is not that good for humans. Okay, so some genes, defective genes will also be there. Now that defective gene also will be expressed where? Into the new plant which is produced. So plant A, plant B, one is high yielding, another one is disease resistant. Now what I am going to produce? I am going to produce plant C. Okay, by crossing these two I am going to produce what? The plant C. So plant C will be what? It will be having both high yielding and disease resistant. Chances are there. Okay, but at the same time this B, if it is having some defective characters, as I told you, it is producing some other poison which is harmful to human kind. So what will happen? Sometimes that also will be expressed. The defective gene also will be expressed. So what I am getting, C will be having high yielding, disease resistant, but it is producing some poison which is not that good for humans. I couldn't remove this one. But with the help of the modern biotechnology, not the traditional hybridization. So what I dealt was traditional hybridization. Like now with modern biotechnology, what we can do? We can just remove that segment, the gene which is coding for that poison. If you are removing it, then automatically what will happen? The one which I am going to get will be only high yielding and disease resistant. Only the desirable gene will be expressed. The undesirable genes will be eliminated by cutting and pasting. Okay, so that is the advantage of what? Genetic engineering over traditional hybridization. Next point is, the first recombinant DNA was constructed by who? Constructed it? Stanley Cohen and Herbert Boyer are the two persons who have constructed the recombinant DNA. rDNA, first rDNA was constructed by these two persons. Okay. The 3 steps have been listed with very simple points. So, for making any genetically modified organism, not the original one, I am going to make a new genetically modified organisms. So, I have to consider some steps. So, what are the different steps? Many are there, short they have given 3 steps. First, identification of DNA with desirable gene. The first step is what? Identification of DNA with desirable gene. Like for example, this is a plant, okay. This plant or this tree is having some good character, like they are producing some rich protein, okay, some protein which is good in nature. So, this protein is being produced by this tree because of a presence of one gene which is coding for that protein, isn't it? So, first I have to identify from the cell, I have to take out the cell of the plant, thereafter I have to study the DNA, then I have to identify in the DNA which is the segment which is coding for the gene which is producing that protein. So, first is identification, okay. So, identifying of DNA with desirable genes. So, I have identified this plant is having this DNA, in that DNA this is the gene which is producing the protein. So, that is the first process. Second, introduction of the identified DNA into the host. So, any organism, any plant I want, that one means what I will do, I will introduce this DNA, I will take it out, cut it out, take it out and introduce this one into the seeds or the embryo, I am going to introduce it. So, that plant will also be having what? The host plant, okay. So, this is the plant which was not having that protein. Now, from these I am collecting the seeds. Into the seeds what I am going to introduce? I am going to introduce this recombinant DNA, means that DNA which I have taken out, I am introducing it into this DNA, okay. So, this one is called as what? Introduction of identified DNA into the host. Next step what the biotechnology have to keep in mind is that, the genetic engineering has to keep in mind is that maintenance of introduced DNA into the host and transfer of DNA into the progeny. Now, this DNA I have introduced, now this seed will be growing and will be producing that protein, right. So, it has to produce the protein. Along with that, this tree will be producing many fruits and they will also have seeds. That seed should also have what? This characteristics. That also has to be taken care, so that next generation I will be getting the one what I have introduced into, because it is very difficult to introduce something. So, after introducing the only one tree is producing, from there the next generation again I have to create a new, it's a headache, isn't it? So, what I have to do is that I have to see to it that the introduced gene is expressed where? In that generation. Along with that, the next generation, the offspring what they are producing should also have what? This introduced gene, right. Now, when you are moving on to tools for recombinant DNA technology, there are different tools. So, what are the different tools? I need for this rDNA technology some materials, some substances are needed. So, what are they? Number one is your restriction enzyme, second one is your polymerase enzyme, third one is your ligase enzyme, fourth is a vector, okay, and fifth is a host organism or cell, okay. Now, this one by one we will be discussing in the process of rDNA technology short today, in detail in the next upcoming classes, right. So, 3 steps in gene engineering. First what? Identification of desired gene. That desired gene is taken and introduced into a plant in the laboratory. They are introducing this gene into the seeds or embryonic stage they are introducing. After introducing, what they are allowing them to grow, so that they are maintaining and expressing that protein is produced or not, they are seeing, as well as the seeds which they are getting out of it. The plant is producing fruits, the seeds they are collecting and then they are checking that whether that is also continued for the next generation. So, these are the steps by which we can make GMO. What is GMO? Genetically modified organism. So, what is the full form for GMO? Genetically modified organism. So, in order to make this GMO, what and all step? Identification of DNA with desired gene, introduction of the identified DNA into a host, maintenance of the introduced DNA into the host and transfer of DNA to the progeny. So, these are the things that has to be kept in mind while making what? A genetically modified organism. because I am making a tree which was not producing that protein. I have introduced that protein from some other tree into this, and that tree is now going to produce that protein. Okay, so that is what genetically modified, not the natural one. I have introduced a rDNA, means I have introduced a foreign DNA into it. Okay, so that one we call it as what, the genetically modified organism, not the original organism. I have modified their genetic makeup, right? So that is what is dealing with, the genetic engineering is dealing with. Okay, rDNA technology. Okay, so with the help of this diagram, I will just give a short explanation regarding what rDNA technology. In detail we will be learning later. Now just to understand, right? Now listen students, foreign DNA, first diagram you see, foreign DNA, gene of interest, desired gene, all are one and the other, name the same thing, it's all denoting the same concept. Okay, foreign DNA. Now listen, so this is a plant. Okay, in this plant is disease resistant to pest. Okay, so what do you mean by pest resistant? Means what? It is resistant to pest. The pest is not attacking the plant. Maybe the pest is some insect, that insect is not attacking the plant because that plant is resistant to pest. Why it is resistant? Because it is producing a toxin. Okay, it is producing something which the insect doesn't like to go near and have it. If the insect senses danger, if some poisonous secretions are there which will kill the insect will not go near that plant. So that secretion which is produced by this plant, when you are seeing, this plant is producing that secretion because of the presence of a gene. That gene is coding for producing that secretion. That secretion is like a toxin, so the insect will not come near the plant, but it is not toxic to us. So in order to make this pest resistant plant, I have to make, so this plant is resistant to pest, but I want in my field all the plant being pest resistant, it has to be resistant to pest. So I am going to make pest resistant plant with the help of rDNA technology, right? Now this pest resistant plant, I told you, is having a gene which is coding for a protein, which is coding for a toxin, which is dangerous to insect. So that toxin producing gene, that one, the gene, that DNA, we call it as the foreign DNA. What do we call? Foreign DNA. That gene is called as gene of interest, because I am interested in that gene. I am going to take out the gene and play with that gene, making pest resistant varieties. Okay, so gene of interest, or we can also call it as desired gene. What we can call it? Desired gene. So foreign DNA, gene of interest or desired gene is that sequences which is producing the protein which is resistant to pest, right? So now what I can do, I have to take out that gene which is producing that protein, okay, or producing that toxin. So in order to take that gene out from the thing, first I have to dissolve the cell wall. If it is a plant cell, obviously plant cell pest resistant we are speaking. So dissolving the cell wall is done, nuclear membrane is dissolved, the DNA is slowly taken out, and thereafter what they are doing, they are adding an enzyme in order to cut those DNA into different segments. Okay, so this is a DNA. So imagine this is a DNA. Now this DNA has to be cut into many segments. Okay, so for cutting it into different segments, I will be adding what? I will be adding restriction enzyme. What I am going to add? Restriction enzyme. Specific sites are there, specific locations are there in the DNA. Correctly, this restriction enzyme will go and cut those segments and make it into fragments. Okay, so that is the role of what? The restriction enzyme. So this is a plant and this plant is disease resistant. So what I have to do? I have to take out the cell from the plant. Then I have to dissolve the cell wall, nuclear membrane. I will take out the DNA from that cell. Okay. After taking out the DNA, I will add restriction enzyme. I will make many fragments of that DNA. In that DNA, I will be selecting, I will be checking, I will be finding out the gene which is coding for that toxin that is taken out. that is the first process. Okay, that segment which is producing that toxin is taken out. What do we call this DNA? We call this DNA as a foreign DNA, gene of interest or desired DNA. So this is the foreign DNA which is taken out from here, right? Now this foreign DNA, what I will do is that immediately I will not put it into the host. Some other plant I will not put it. First I have to make many copies because it's a very tedious process. If I am taking only one and directly putting it, what will happen? Sometimes I may fail, isn't it? But the process will not take place. So it will all go waste. How much effort we have taken in dissolving the cell wall, nuclear membrane, then taking out the segments, gene has been isolated. So it's very difficult task. So this foreign DNA, immediately after taking out this foreign DNA, what we are going to do is that I am going to choose a vector. What I am going to choose? I am going to choose a vector. Okay. So this vector is a bacteria. So this is a vector. Who is a vector? This one is a vector. And what is this? It is a bacteria. Bacteria, when you are seeing this bacteria, you have learned the structure of bacteria. The bacteria is having its own DNA plus an extra chromosomal DNA. One more DNA, it is present beside the main DNA. That extra chromosomal DNA, we call it as plasmid, not plastid, it's different, chloroplast, right? So it's plasmid. What do we call them? Plasmid. So extra chromosomal circular DNA is present. That one we call it as what? We call it as plasmid. So second step, what I am going to do, I am going to take a bacteria. From that bacteria, I am going to take out the plasmid. What we are going to take out? The red round you can find, right? So that red round is what? That red round is the plasmid. So that plasmid is taken out, and in that plasmid also the same restriction enzyme is put. What is put? The same restriction enzyme. So how to take out that plasmid? You know, I will dissolve the cell wall. Then nuclear content and all will be dissolved, means the cellular content will be dissolved correctly. I will be taking out that plasmid. In that plasmid I will add what? Restriction enzyme. That plasmid will break. Okay, segment will be created, a fragment will be created with the help of what? Restriction enzyme. I am creating that segment, and into that segment what I am going to introduce, when I am cutting with one particular restriction enzyme, one segment will open. Okay, so in that segment what I am going to introduce? I am going to introduce this foreign DNA. Understood? So what is this foreign DNA? It is pest resistant. It is coding for pest resistant. It is having a gene which is producing a toxin which is resistant to pest. Okay, yeah. So what has happened? Now I have introduced my foreign DNA into the plasmid. Clear? After introducing this DNA, so this restriction enzyme I have added, then only I have cut that one, introduced it. Okay, so this origin of replication means this DNA should multiply and produce many copies, right? So it has to replicate. Replicate means what? So from one DNA I have introduced, so this is a circular DNA. So imagine this is a circular DNA. Okay, so in that what I have taken out? I have taken out one segment. In that segment I have introduced what? I have introduced I have introduced my gene of interest, right? So the foreign DNA is this blue color. Now this plasmid should have one site. It should have one site called as ORI. ORI means what? From one, origin of replication. What is replication? You know. So this is one circular DNA. From this, next how many should be produced? 2 should be produced. From 2, how many should be produced? 4. So what is happening actually? The DNA it's unwinding, you know. Yes, they will move. This will produce one another DNA. This one will produce another DNA. Like that, many copies has to be produced. So this replication has to take place, means that only with one it's not enough. Many plasmid should be produced. In order to produce many plasmid, I will be choosing a plasmid which is having ORI site. What is the site? ORI site. What is ORI? Origin of replication. If ORI site is there, then only from that plasmid you can find many copies are produced. If it is not there, then automatically what not will be produced? Many plasmid will not be produced. Only one plasmid, not, it's difficult, right? So ORI site should be there. Next is what? Polymerase enzyme also I have to put into it. So for this process I also have to introduce what? Polymerase enzyme. What is this polymerase enzyme? They will synthesize. This DNA has to be synthesized, right? So this DNA, circular DNA 1 to 2, 2 to 4, how does it happen? Because of synthesis. Synthesis of DNA is taking place. Who is helping in this synthesis? Polymerase enzyme. So what is the role of polymerase enzyme? It is helping in synthesis of DNA. Now another thing is ligase. What is the enzyme? It is again another enzyme. Why it is needed? I told you, right, what we are doing. This is a circular DNA. So you can find the red was what? A circular DNA. In that red circular DNA I have cut one portion, right? After cutting one portion, in that what I have introduced? I have introduced my foreign gene. So this gene, when I am introducing, whether it has to be joined with the original portion or not? Yes, it has to get joined, isn't it? Then only they can divide. So for joining these thing, we are using another enzyme called as what? Ligase, right? Now I will make a bit clear. What I have taken a foreign DNA. This foreign DNA is being isolated. Now I am taking a bacteria. In that bacteria there is an extra chromosomal DNA called as plasmid. This plasmid should have some property. What is the property? It should have a site, a region where the replication process starts. So that site we call it as what? The ORI site, origin of replication. What is replication? One DNA converted into two DNAs. Okay. So how it is taking place? One DNA is unwinding. They are producing its duplicate copy. This one is producing its duplicate. Like that two. For that what is needed? ORI site. Now next is what? Restriction enzyme. What is the role of restriction enzyme? They will go and cut at specific locations. So for cutting the DNA, what is needed? Restriction enzyme. ORI site I told you. What is polymerase enzyme? It is used for synthesis of DNA. What is the role of ligase? They are helping in joining the foreign DNA with the plasmid. Okay, that is the function of what? The ligase. So hope this is clear till now, right? Now what I have got? I have got a foreign DNA with the plasmid. Now what I am going to do is that this DNA is introduced. I am taking out a bacteria, another one more bacteria. Into that this plasmid is introduced. The recombinant plasmid is introduced. So this one is rDNA. Why? Because it is having a foreign DNA along with the normal one, isn't it? So this plasmid is introduced into the another bacteria by microinjection. I am just injecting it into the plasmid, bacteria. Now this plasmid is introduced with the foreign DNA. Now what this plasmid will do? They will multiply. I told you they are having what site? ORI site. They will multiply. They will produce innumerable number of plasmids. All these plasmids will be having that DNA which is coding for what? That toxin which is resistant to pest. Like this many plasmids will be produced. This process we call it as gene cloning. What do we call them? Gene cloning. So this plasmid, when you are seeing after many copies has been produced, they are secreted out and these plasmids are introduced where? Into the embryonic seeds they are introducing it. Okay. So when they are introducing this one, so seed, when the zygote is formed, immediately you can find this again cut and paste only. So with that plasmid is introduced into the seed. Now what will happen? When the seeds multiply, the plasmid also multiplies, and thereafter you can find the host organism is producing what? Resistant to pest. The gene which is resistant to pest is expressed where? Into the host organism. So this is a plant. In that seed of the plant only what was produced? This plasmid was introduced. Okay. So this plasmid is now introduced into that plant, and that plant will be what? Disease resistant. So like that many clones are made, and thereafter that are introduced into the seeds of different plants. So all plants will be having what? Resistant to pest, right? So here you have understood what are the different tools which is used in biotechnology. I told you restriction enzyme is for cutting, cutting at specific location. ORI site, origin of replication. Polymerase for synthesis of DNA. Ligase for joining DNA. What is plasmid? The bacteria which we are using. The plasmid which is used for carrying the foreign DNA from one tree into others. Who is the one who is carrying it? The vector. And who is the vector here? The bacteria with the plasmid. Main one is what? The plasmid is carrying it, isn't it? So that is a vector, right? So with this only tools of biotechnology, recombinant DNA technology will become clear. I need a restriction enzyme, I need polymerase enzyme, I need ligase enzyme, I need a vector which is a bacteria with a plasmid, I need a host organism into which I am going to introduce that one, we call it as a host, seeds of other plants, right. So like this I can develop many varieties by cutting and pasting, okay, using a vector. So this process we call it as what? rDNA technology. Hope it is clear. We will be discussing in detail each and every steps in the upcoming classes. Thank you.